Okay, so can everybody out in Zoom land hear us? Can you unmute and say something if you can hear us?
Yes, I can hear.
Okay, so we are ready to go. So it is definitely my pleasure to introduce our speaker today, Dr. Sally Mackenzie, who is relatively recent to Penn State and is doing some very, very interesting work, among other things, trying to get us some new greenhouses, which would be very, very nice. So with that, Sally, I'll just turn it over to you.
Thank you. So what I want to do today is tell you first some of the work that that my group does here at Penn State, but then I want to put this in the context of some work that I'm hoping will be more feasible on this campus, since I wear two hats. The work that I'm involved in is associated with both the Department of Biology, where I'm thinking a lot about epigenetics and the molecular basis of epigenetics. Department of Plant Science, where we think about how do we capture value from what we study to putting it into the field in meaningful and practicable ways, maybe even commercial ways. And then wearing a hat as the director for the Plant Institute, where we think on a broader scale about overall, what do we need at Penn State to allow us to sort of go to the next level in terms of plant science generally across disciplines, meaning how do we meet the needs of the plant science and the ag college, but at the same time, start thinking about integrating with ag engineering, start thinking about integrating with information science, think about integrating with biology and other fundamental sciences, and whether we can create some sort of space that will allow that. So I want to first just talk a little bit about some of the work that my group does. And that is in the implementing of epigenetic variation for crop performance. And so, when we think about the way that plants sense their environment, we have to think about the fact that we've spent an awful lot of time developing our own sensors. And we need to do more of that. We need to try to understand what plants are seeing at any given time, the dynamics of what a plant sees. Can we measure temperature? Can we measure moisture more accurately? Can we measure light more accurately? We spend a lot of time, particularly in engineering and information sciences, and figuring out how to use this information. But what we haven't spent quite as much time thinking about is how the plant senses all of those things. In other words, we spend our time integrating data sets to understand what a plant is likely seeing in the environment we can measure. But it should also be possible for us to integrate the gene networks that a plant actually uses when it sees changes in its environment and it responds. So we think of that right now in relatively simple terms. Well, we'll just go in and we'll see how genes change expression. We'll try to learn as much about that as we can. But the fundamental problems, of course, are that to be as sensitive as you can in understanding what a plant is seeing means you almost have to go to the single cell level, and that becomes exponentially more difficult to integrate. And if you're not going to do it at the single cell level, then you have to have ways of understanding plant communication that are particularly sensitive, so that that you're, if you will, putting your finger on the pulse of what really the plant is doing, right? You're not paying attention to extraneous data that are leading you astray, but that you're really narrowing in on this is the actionable part of what a plant is seeing and operating on. So we have been, querying those questions or querying those ideas for some time. And it's brought us to the idea of recognizing that plants sense their environment. In one of the ways a plant senses its environment is through its organelles, namely its mitochondria and plastids. There may be other venues, but mitochondria and plastids are important to this. And it's probably no accident that those are also the energy centers for the plant. And that's because as a plant senses changes in its environment, that becomes a bioenergetic decision for the plant, or let's say dilemma for the plant. Because a plant always has, if you think teleologically, the decision to make at a cellular level of, if I'm under stress, how much energy am I going to put into growth and trying to outcompete my neighbors? And how much energy am I going to put into hunkering down and getting through this stress? And if I hunker down through this stress, should I try to race it by basically putting out my seed as quickly as possible? Voila, I survived, even though I may not look very, very good while doing it. Or do I hunker down, slow my growth, and just wait for this stress to be over? Those are all the, the decision points or the pivot points, if you will, that a plant has to make. So I'm going to start with more or less the model of what I'm going to try to persuade you is probably going on. So I'm kind of giving you the end of my story first. And that is that we have discovered, or we think we've discovered, a new organelle type. It's a plastid. But, if you think about the way that chloroplasts develop, chloroplast is a general term we use, but chloro means something specifically. It's going to increase in chlorophyll, and it's basically going to be responsible for photosynthesis. And where that function really goes on is in the leaf mesophyll. That is the business end of photosynthesis. But you have an epidermis, and you have vascular parenchyma that surrounds the circulatory system of the plant. You've also got things going on during reproduction and in the meristem that we know are not photosynthesis. Those are not photosynthetic centers, but they have organelles, plastids. And specifically, those plastids are about a third the size of a normal functional mesophyll chloroplast, and they actually seem to be involved in stress sensing and signaling, signal transduction. So here's the thing. When you look up at this plastid and the mitochondria that it interacts with, we know now, they have proteins that specifically localize to that plastid that you won't find in a mesophyll chloroplast. And some of those are stress responsive proteins. In other words, they will change their expression when a plant sees stress. In addition to that, we know that those plastids have the ability to communicate with the nucleus through reactive oxygen changes and redox changes and through calcium signaling. What we also know is that there's retrograde signaling that goes on with those plastids, meaning that the nucleus can talk back to the plastid as well. So we've taken apart some of the basic features that say this plastid is special. And so now I'm talking about what we call sensory plastids. They look like chloroplasts, but about a third the size. Their thylakoid membrane is not as well developed, and they sit in specific places of the plant, namely the epidermis, the vascular parenchyma, which means the live cells around the vascular tissue, the reproductive tissues, and I can't be more specific yet because I don't know exactly which ones, and in the meristem, which means the root meristem, the apical meristem. Okay, so then the question becomes, let's say that there is a plastid. What could it actually say to the nucleus, and how might the nucleus respond? So I will tell you, and I'm going to show you evidence today to suggest that the nucleus knows how to respond to those signals. And in fact, there are very specific epigenetic responses that occur. And by epigenetic responses, what I mean is that without the plant changing its genes, it has the capability of reprogramming its gene expression. And one of the key networks that it changes happens to do with this TOR SNRK that I'm showing up there, because that happens to be the business end of where a plant pivots to say, do I put energy into growth and out competing, or do I put energy into stress response? And that stress growth dilemma is really an antagonistic relationship. You can't do both. So there have to be decisions made on how I'm going to tweak that, or I'm going to pivot one way or the other. And I'm going to postulate right now that that's what I think epigenetic phenotypic plasticity, response to the environment, does at a very cellular level. That's the area in which these decisions are being made. So let me just tell you all the reasons why I believe that. So our lab works with one of the genes that is sensory plastid specific. It's a nuclear gene, but it only expresses, as far as we can tell, and accumulates its protein in those tissues I told you about, the epidermis and the vascular parenchyma and the reproductive tissues. And I'm just showing you that here, that basically if you were to look at where you see accumulation, and here what we're looking at is just taking that gene and fusing it to a reporter so that you're able to, see where the protein is accumulating. And here with Gus staining, you can see it's in the epidermis, the vascular tissue, the meristem, and the reproductive tissue, as I mentioned. And that's how we first discovered this rather specialized expression. But there are other things. Let's say that we think that MSH1 is a part of this phenomenon. And in fact, I do. I think that MSH1 is one of the components of the plant's ability to see stress, change MSH1 expression. That talks to the nucleus, and it makes a change in the way the nucleus behaves so that the plant reprograms its growth. That's the model we're building. Then one of the things we have to say is, how on earth could the presence of one protein in a plastid have all of that effect? So what we know is that MSH1 is a gene that has basically a DNA binding protein, or a DNA binding domain. And we know that that protein, when it's expressed, can accumulate not just in the plastid, but also in the mitochondrion, in those two energy centering parts of the cell. And we know that because we use something called hemicomplementation, which is a really simple genetic way of taking a protein that's normally dual targeted, taking off its targeting pre-sequence, replacing it with a pre-sequence that only targets the protein to the mitochondrion or only to the plastid, and then saying, okay, if it only goes one or the other, I've complemented that one, and I'm looking at the effect of depleting the other. So when the plastid does not have MSH1 expressed, we see an effect. We don't see it when the mitochondrion doesn't have that effect. So we can use genetics to actually figure out where is epigenetic signaling occurring. But what we also know is we can knock out just that DNA binding domain specifically. And when we knock that out, we know that we create that whole signaling change, that whole change that reprograms gene expression. So that says that the ability of the protein to bind to DNA in the plastid is what's important. And if we pull that gene or that protein out, then it destabilizes the genome. And by destabilizing the genome, we create our signal. And then that signal is sensed in the nucleus. And then we think all these other things will happen. So we've now at least started to delimit a part of how this could work. Plants see a change. And if that change is severe, MSH1 as one of the signalers will actually change its expression. And by doing that, causes this cascade downstream that allows the plant to go, oh, I'm in trouble. I need to change the way I grow. So this is basically the way the system works. And this system is interesting because it's been around all the time and we've never actually noticed it. In other words, it's been under our noses, and probably breeders and growers have seen signs of this for a long time. But the people who've really picked up on the signs have been the ecologists out in natural populations. And the reason for that is because they're often looking at things that grow in the understory of a forest where some are in high light and the members of their same population are in the shade, and they can actually look at how the high light and the shade guys interact with one another. So they already know a lot about how plants can adapt to their environment. Whereas we, as agricultural scientists, give such a uniform environment, we never actually can see these things more or less in a single population. But if you were in a Arabidopsis to start with, say, a wild type plant, and we were to artificially downregulate MSH1 the way that stress would do, we can create all kinds of changes in the way the plant grows. And we've seen this if you work in corn. There are certain lines of corn, WF9, for example, where you can walk out in a field after a terrible drought and you get these funky looking plants. I mean, they're just really looking odd. If you take progeny from those plants, likely you're going to find changes in the way the progeny grow. But of course, we always rogue them. In this case, if you were to take the progeny from those plants where MSH1 is downregulated, they will have memory of the stress. Some of them we call non-memory because you can't see the effects. But those with real strong memory, you see the effects. They're slower in growth and they're very heightened in stress. They're very drought tolerant. They're high light tolerant. They're really altered in the way they're expressing their genes, but we've done genetically nothing to them. We went in and temporarily downregulated a gene, and then we came back out and restored it. But the next generation remembers it. And generations after that remember it. And generations after that remember it. So this is a heritable memory of something that happened to a parental type. And then if you were to take either the stress or the MSH1 mutant, something where MSH1 wasn't functioning, and you cross it, or you graft it to the same genotype. In other words, we're not making changes between different varieties. This is all the same genotype. Then what you'd have is when you graft it, you use your MSH1 mutant as your rootstock, and then you graft to it wild type. Or you can use your MSH1 mutant as a female or the male in the cross and cross it to wild type. In either case, you'll get enhanced growth, enhanced vigor, enhanced rate of growth, and enhanced yield, and enhanced resilience, kind of like heterosis. But we haven't created heterosis. Everybody following? So realistically, we've created state 1 as the mutant or the downregulated MSH1, state 2 as the memory, state 3 going through grafting, and state 4 as going through crossing. All of those are different growth states that all came from the same place. They all started with Columbia 0. So here's the thing about this. There are a lot of different things that can happen. And let me say, some of these phenotypes can be quite remarkable. So we know we're altering lots of pathways. If we grow an MSH1 mutant at short day length, some of those plants will actually become perennial, woody, and growing 7 months with leaves and flowers. Really unusual pathways are altered. So these aren't minor changes. They're fairly major to the way this plant grows. But even more important than that, we can recapitulate everything I described to you in any crop we've tried so far. So that means that if I take a tomato plant and I downregulate MSH1, I will likewise get memory. I will also, when I cross it back to wild type, get enhanced growth and vigor. And I will also, if I graft it, see in its progeny enhanced growth and vigor. Any plant species that we've tried, monocot or dicot, understands this. Now, of course, with monocots, you can't really graft them, but they go through the same process. So this is a language that's conserved, at least among flowering plants. And more importantly, this is a language that's epigenetic. So in plants, in plants, there's been a lot of work done on what the epigenetic communication system is like. In other words, how is it that plants can reprogram their expression? And one of the methods they use is called RNA-directed DNA methylation. And RNA-directed DNA methylation means what it sounds like. They're small RNAs that identify the target sites for differential methylation. And it turns out that in some species, those small RNAs can be inherited, okay? The RDDM pathway has its own components. And so we can use various genes that we know are integral and start testing them in our system. So for example, we know that if you want to go from wild type to the MSH1 mutant, then a histone deacetylase 6 has to be present. If it's non-functional, this whole process breaks down and that's lethal. So what does that mean? It means that already, just to create these changes, the underpinning of that histone deacetylase has to be there. That capacity has to be there, or MSH1 can't do its job. We also know a second gene, DRM2, which is a methyltransferase in the RDDM pathway, has to be there, or you'll never see memory. We also know that, for example, when you do your grafting, where you're taking a bottom wild type and putting on it a bottom MSH1 mutant and putting on it a wild type scion, that there are small RNAs that have to move through that graft into the progeny to make those changes. And we know that because if we use dicer-like 2, 3, and 4 mutants together in that rootstock with MSH1, we no longer get any of these effects, including the methylation changes that would happen in the progeny. So we can actually dissect this process genetically and learn how epigenetics alters growth in plants, either for stress or for enhanced growth, for that decision point. And it turns out that we can even contrast the outcomes that come when we have our mutant and when we don't have our mutant. And by contrasting those outcomes, you can look at what methylation changes happened when we didn't add those mutants, and then what methylation changes did we lose when we did add those mutants. And that allows you to look at exactly the targets that Mother Nature is using to say, we're going to enhance growth now. This is where we're going to go. Because you can do this direct contrast with and without those mutations in order to see what changed. So those make for very powerful data sets. And the reason is because they're reproducible and because they're really plus and minus gain. They're very qualitative. And that's what you're looking for in a system that can be this noisy. So for example, we can learn what some of these components do. HDA6 has already been shown to control the circadian clock pathway. Circadian clock is really important in plants. It defines when flowering is going to happen. It defines how growth is going to occur in short versus long day, when a plant is going to become dormant, and so on. And it turns out that in that MSH1 HDA6 double mutant, where I said it all breaks down, what I didn't tell you is it only breaks down if I'm growing those plants at 12-hour day length. But if I move them to long day, to more like a summertime, then in fact, what happens is it can survive. So what that tells us is that these changes for environmental sensing are actually dependent on the circadian pathway. And it detects what we're looking at in short day versus long day, and that influences its ability to continue. So putting that all in kind of a nutshell, what I think that means is that there are lots of things that happen to plants during their development, during their response to environment, just in maintaining their genome. And all those things have to be put right, if you will. In other words, plants can be disrupted a lot, but you have to reestablish homeostasis, some kind of normalcy afterwards. And I think that HDA6 participates in that realignment, getting things back to normal. And so if you don't have HDA6, when we create this disruption with our mutant, then in fact, the whole system breaks down. But when you have HDA6 there, it has the capability of realigning, and in this case, deacetylating chromatin to restabilize those regions that we've been disrupting. So the reason I go into this is to say, you can study a system like this on the very, very fundamental end, and it actually informs then the way that you want to deploy this in the field. And I'm only putting this slide up to say there's not only one gene involved. This is PPD3. It's also associated with the sensory plastid. It also gives rise to memory. And I won't go into all the details of this again now, but you can see that in this case, we get very, very small and very, very large plants out of this. Again, that decision of do I hunker down with stress or do I try to outgrow and outcompete our competitors? So the two ways we've perturbed the system, we get that same decision point of do I grow or do I hunker down and deal with stress? And we can even go so far as to figure out where these are in the sensory plastid. We use floors up above to actually see them. And then we use electron microscopy to localize them. And over on your left, I'm showing you where MSH1 resides, sitting inside the nucleoid where it's binding to DNA. Remember, it's a DNA binding protein, and it destabilizes the genome to cause the signal. But in PPD3, it sits in what we actually think, you can't see it here, is a plastoglobule that's sitting up right next to the nucleoid. And it also sits out on the thylakoid membrane. And that's because PPD3 has another day job that is not DNA binding. So these are two proteins that both can influence the way sensory plastids behave, but they're distinct from one another and may work coordinately. And when we do this work, we always know we're looking at the sensory plastid because its size is so different from its neighboring mesophyll chloroplast in another cell. So we're actually describing a totally new organelle that people haven't thought about before, and it has very distinct features. It turns out that some of these proteins, like PPD3, seem to actually decide what a plant is going to do at the meristem level. So you can see over on the left, if we disrupt these organelles with an MSH1 mutant, then PPD3, now that we can see it with GFP, actually is upregulated. And you see lots of concentration down at the tip of the root over there on the far left side of the screen, because when this is disrupted, PPD3 is actually changing the way those organelles behave. You can also see in that mutation that if we downregulate or knock out PPD3, we change the way the meristem develops. So what that tells us is, just like I was telling you with MSH1, if you knock out things that make the genome unstable, it makes a signal. Those things must happen in the meristem. A lot of the business end of how you define growth seems to be happening very, very early. And that's where these proteins seem to be functional. So when we work with mutants, and when we spend time thinking about where in a plant these things might be operating, it helps you to model the way you think this ought to be working in the field. It's how early is it going to be, what tissues are going to be important to it. And therefore, if you ever wanted to develop an assay, is it working, is it not, you know what tissues to be looking for. So there's value in combining this fundamental information with practical information like this. Over on the left, you're just looking at one stress tolerance phenotype, which is response to drought. And as you can see on the far right of that, MSH1 memory is by far the most drought hardy, but PPD3 overexpression also shows really good drought hardiness after 32 days of non-watering. And we can even start to model these things. Up in the left, you're looking at when a plant sees stress. MSH1 is associated with the thylakoid membrane. It actually will cause MSH1 to downregulate. Now you get genome stability, and then you get a redox change. And that redox change is associated with PPD3. And on the lower left-hand corner, you can see that we know that plastoquinone is altered in its redox status. So we know that there are things we can measure that tell us these organelles are altered. And organelles are very interesting. When they're stressed like this, sensory plastids will move right up to the nucleus and actually sidle up to them, cuddle up to them, and they put out these things called stromules. They're almost like arms reaching out to the nucleus and actually contacting the nuclear envelope. So it's pretty cool. I mean, in real time, you could watch a plant see stress and you'd watch these little organelles just move right up to the nucleus. And that is what's thought to start this communication. And in the end, the way the nucleus seems to respond is by either creating memory through MSH1 in the dwarf, or enhanced growth through crossing, or with PPD3 to make the dwarf and the enhanced growth all in one population. Either way, these plants seem to know that they have to make that decision once they've seen stress, and we're actually watching it in real time. And so now I just want to very briefly say some of the methods that you can use to understand in detail how this works. You can use very specialized methods for characterizing DNA methylation patterns. You can sequence the whole genome, but you can sequence it using bisulfite sequencing and identify where cytosine methylation comes and goes. You can then use systems like the MSH1 system where we can derive these different epigenetic states, and we can use mutants to basically change these populations. And then you can use basically protein-protein interaction network analysis, and that allows you to go in and say, what is the network that's being targeted? What genes are talking to which genes? And which are the central hubs that seem to be really the vital targets to making a plant really change the way that it grows. So we used to think of things in a single gene way. Now we have to think of things in a network way in order to understand how whole phenotypes will change. And in a nutshell, the way that we do this is kind of nice for our system because it's very difficult to resolve those kinds of data. But we can actually take those 4 states that we create of epigenetic reprogramming, look at their differentially methylated genes, only the genes that undergo change, and then see what they have in common. We find those 871 that are shared. That means that whenever MSH1 is involved, those 871 undergo change. And then interestingly, we can say, but did they undergo different change? So out of those 871, you can use what's called hierarchical clustering using, and I won't go into the details of this, but you're actually just looking at the way that methylation changes happen. So let's take an individual gene and say, if methylation change happened when it saw MSH1 mutation, is that different from the way methylation happened when it saw memory? Is that different from the way it happened when we grafted? And is that different from when we made our cross? And what we can do with those different patterns is we can now actually do this clustering that says that the answer is yes. That when you look at wild type in controls, you can discriminate them from each of those 4 states, just looking at methylation of those 871 genes. So that means not only are those 871 targets, but it means that they were targeted in different ways if the plant decided to grow and if the plant decided to hunker down for stress. And this is kind of what you'd see as a readout. Over on the right, you can see that we're looking at changes that happen with one kind of mutation with PPD3, changes that happen with different types of MSH1. And what you're looking at is changes in cytosine methylation along that particular targeted gene. Everything in red is a CG methylation, and then blue and green give you different contexts. And you can actually look at the height and whether it went up or whether it went down from the line. And that actually is a readout of looking in really high detail at what happened within a gene in response to the changes that we made. If it went into memory or if it went into one of those other states. This is a lot of detail. It's a lot more detail than in general we've ever worked on in plants before to understand epigenetic change. And what it gives us is the ability to now see who are the players. And so what it gives you, you're basically looking at the key players out of the 871. And what I mean by that is only 531 are actually already known to be in networks. We don't understand the others yet well enough. What I want to point out is I have them in red and blue. Everything that's in red, we know to be what we call an RDDM target, meaning an epigenetic target. And we know that because remember, when we graft, we can graft with mutants or without mutants and see that on and off for changes in methylation. So we can overlay those data sets with this and be able to say everything in red, we already know genetically is targeted for epigenetic change. That's a huge amount of information. And more importantly, what you can also say, now 77% of these are known to be RDDM targets. So these are the core of the MSH1 effect shared by all of those states. And they're also the genes that undergo differential changes depending on what state we're looking at. And so what you can do is you can cluster these. And the way you do that is by a method called K-means clustering. And that allows you to look at who are the genes with the greatest centrality and connectivity, meaning those that appear to be most likely hubs of the gene networks we identify, meaning that if you were to pull them out, the whole network would fall apart. They really are central to that hub. And those turn out to be 67 genes of the ones we've identified that are key hubs. Those that are in red, again, we know them to be RDDM targets, but I want to point out that the beauty of a system like this is that you can identify now the key players in response to stress, the key players in development or growth, the key players that we believe in gene expression, which is what we're altering. Spliceosome means that we're altering some of these by alternative splicing. And then histone modification and chromosome organization in the upper left are because, of course, this is an epigenetic change. We're changing methylation, we're changing small RNAs, and more importantly, we're changing histone behaviors. What I want to point out is there are some guys that really stand out here and have been very informative. So now we're really talking about the key deciders in a plant's decision for how to respond to its environment. One of them is splayed, and that's a gene that actually acts to identify meristem identity. And so that's one of those early players that decides in the meristem what's going to happen. TOR is one of those genes that makes that decision. Am I going to hunker down or am I going to grow and try to outcompete? And that pivot point happens at TOR and is known to occur at TOR. Another is this NRPV1, which is basically the largest subunit of RNA polymerase II, a key regulator in gene expression overall. The next one is basically a gene that is specific for the RDDM pathway and also interacts with that histone deacetylase 6 that I was mentioning earlier. And another one is a key component of RNA polymerase IV, which regulates the RDDM pathway. So all of these fit very nicely into the first story I told you, because these are the key regulators of everything that we know and have seen occurring within our system. So I'm not going to go into detail, but to say, just to show you this and say, a gene like TOR is really central. These are just a few of the interactions that TOR as a single gene mediates. So when you start messing with something like that, you're messing with a lot of different components at one time. And this is the value of looking at network analysis in plants compared to just single mutation analysis. is it gives you a much broader understanding of how do plants sense their environment, and through some key regulators have the ability to really alter their growth with not too many changes. So here's how we think things work. You start with sensory plastid signaling. The plastids recognize changes in light or temperature or in moisture levels or general stress responses. They have the ability to speak through either MSH1 or through PPD3. Those are two signaling molecules. In one case, creating DNA instability, in the other, creating redox changes that operate at the meristem to give rise to changes at really key targets that will influence growth and stress response, chromatin reorganization, and alternative splicing to give you phenotype changes that make sense. So here's the thing, that whole system now can be transported into and implemented in canola, in tomato and tomato hybrids, into sorghum, and into soybean. In all of those, we actually have field data that has allowed us to now try to implement these, even to move them into quasi-commercial varieties to ask, what would actually happen if we actually put a system in place here? And the question becomes, do plants normally show you this much of their phenotype when you put them out in the field? But do they have this much phenotype to play with? In other words, we can only recognize this by the way we develop our genetically developed varieties, but they have a certain plasticity in their phenotype that we can't access. So what if we were to breed not only the genotype we want, but superimpose on that the epigenetic plasticity that we want? Do we have the ability to make plants more resilient, more able to adjust to their environment? And we're trying that in all of these 4 crops using the system. The way that it's implemented is that basically you use this very simply. If I want to do this in canola, I take their best variety. And now I introduce to it either a transgene or a mutation or a CRISPR mutation that would downregulate or knock out MSH1. The next generation, I would now take that mutant and I would use it as rootstock. As soon as I know that it's working, I use that as rootstock and I graft to it the wild type. So I don't change anything about their variety. I collect seed from that graft, and now we start implementing it in the field. It's that simple. So literally graduate students can implement this. And in fact, we've had graduate students implement this. You know, it's a simple method in the lab. Then you take it to the field and then you ask, now what do we see? And then in the field, you're asking, okay, so it's good in, let's say, generation 2. What about generation 3? Does it hold? What about generation 4? What if we cross it and use it in a hybrid configuration? These are all the different ways that we've attempted to do this. Or in sorghum, what if you can only cross but not graft? How much harder is it? How much variation do you get? Does this work? In soybean, we've seen somewhere on average of 8 to 10% yield increase through grafting. That doesn't sound like a lot, but for soybean, that's huge, especially if you can consistently do that with commercial cultivars, because it's simple enough that as you improve your commercial cultivars, you can superimpose this again and again. 20–30% yield increases in epi lines of sorghum, but there we also have tolerance to cold. And that's important in sorghum because often you have to grow in early times because you're almost always growing sorghum in marginal lands. And you're also going to be growing sorghum in places where you're likely to see drought fairly late in season. So you need that tolerance to cold to get in early. We also see tolerance to drought, and we've also seen tolerance to very low nitrogen levels. Those you might see, for instance, in certain parts of Africa that are truly marginal. 8% are 8 parts per million nitrogen as opposed to the normal 30 parts per million that you would see in the Midwest. In tomato, we've seen 20 to 25% yield increases in hybrids through grafting, and we've also seen tolerance to heat down in Florida. In Chile, 2020, we basically grew these, developed open pollinated and crossed materials, and then took them to North Dakota this year. We just got our data from North Dakota, and actually it looks pretty good because North Dakota got hit harder than any place in the country this year for drought. And we were able to see about 5 to 10% increases in yield under variable conditions, where I mean they're very extreme conditions. Like I said, most growers actually saw about 20% of the yields they'd normally see this year. So we had a real extreme environment in which to test these. And these are the people up above that basically did the work that I just described to you, together with some of our collaborators, Mike Axtell, and then some people from Epicrop Technologies, which is a small startup that allows us to do our field trials. And with that, what I want to now say is that what this has done as well is to help inspire me to think a lot more about what we need overall at Penn State in terms of growing our capabilities. It's not just that our greenhouses really need to be remodeled, although they do, and they will be. It is that we have to start thinking much more holistically. What I've tried to impress upon you is the value of thinking about networks, not just individual genes, the value of thinking about introducing traits to more than one species so you can actually test how common is a certain language or a certain phenomenon. And the value of sort of thinking outside of the box. How can we actually take things that we learn at a really fundamental level in the lab and then moving them into true field conditions? All of those things take serious cooperation on campus. And in my mind, that serious cooperation is going to have to extend not just from plant science as we think of it related to crops to, let's say, forest science and ecology, but also as we think about how it's going to relate to the way we're going to move into new environments, like, for instance, vertical agriculture. Can we take, for instance, epigenetics and make plants happy to live under artificial light and under, kind of nuanced irrigation system that would be really not, as natural as what they normally see? I don't know, but how are we going to ask that question when we don't have the ability to do vertical agriculture systems on campus? We need ag engineering to come in. We also need climatology to come in. We need a place where all of these kinds of features can work together to think about what clever ways do we have and nuanced ways to understand the way plants will see new environments as we deal with climate change, but also how we're going to transport that to other environments. So with that, we've started the process that we call PlantWorks. And the PlantWorks project is one that involves all of what I've described to you. What PlantWorks is right now is it started out as a greenhouse renovation project. Then it turned into a greenhouse renovation plus phenomics, plus plant transformation center. It's also moved into, because we don't have CRISPR technologies on campus without a serious plant transformation capability. It's also moved into a home for safes so that we can start thinking about sustainable agriculture, but in a meaningful way, and a home for ag engineering to interface with information science with plant science students, so that students are basically trained to think on multiple scales. And at this point, it has now reached Nick Jones's office, and it has now started to become a priority project for the campus. So what I do with that right now is to sort of spread the word so that everybody's familiar with what PlantWorks is supposed to be. And I spread that word to administrators. I spread that word to people like Nick Jones or to Laura Weiss. And I spread that word now, next phase, to OPP, where we do a feasibility study. And that happens in the next few months. That will initiate. And if we're lucky and that initiates in a full scale, then that means that the Plant Institute then turns all of their energies to basically talking outside of Penn State. We talk, I'm imagining that our next phase will be to go outside and talk to stakeholders, particularly those who care about the forests around us. We also are going to be talking to stakeholders who care about our being able to use a lot of technologies in the field and also in the vertical ag space, in the urban agriculture space, which is really driving distance from us. It also means that we have to start thinking about stakeholders further outside, those who really want to see new and innovative crop technologies, maybe even like the ones I'm describing to you here, things that really are outside of the box thinking, but that we can actually move along to the field as quickly as possible on a larger scale. So, the two hats that I wear is that on my lab scale, I'm constantly thinking, how can we do something in the lab? And then think about what its implications might be to the field. But at the same time, we also all need to start thinking, if I wanted to take the research that I have right now and really think on a grander scale, what does Penn State need to offer me in terms of an environment that allows me to take that next step? And so as you have ideas, as anybody online has ideas for things that really have to be part of PlantWorks, I'm all ears. And I meet with an executive committee. I meet with a whole host of people that and respect, who are basically trying to assimilate these ideas into, for instance, one of the things that I didn't mention, but one of the things we cannot do right now is to work with proper containment facilities. For some of the insects and or pathogens that are most dangerous to our forests as well as to our agricultural system, that's one of the chief concerns that we've got in the East Coast, because that's where climate change hits us hard, is that we've got all kinds of things flying in here that we can't really control or even understand biologically. They're very tricky to study with the facilities we have now. So that's all a part of the plan. It wasn't initially, it is now. As you think, yourself about things that we really need, and particularly in student training space. Right now, we imagine new places where students who are in plant science interact with and learn from ag engineering students, learn from information science students. What can I do with this data other than what I'm doing right now? And how do I integrate that data into things that I can take, as actionable, outputs from my work? Those are the kinds of ways we need our students to think for next generation. Imagine 30 years from now, you're never doing a laboratory experience. You're only developing things from data already available. How many of our students feel equipped to work in that level of data science as opposed to as it relates to agriculture? We're not quite there, but we can be. So those are all the things that we're trying to integrate into the let's say, teaching, training, and research space that soon will be PlantWorks. I'm hopeful, I'm cautiously optimistic that Penn State administrators recognize the urgency. I'm also cautiously optimistic that the Biden administration recognizes the urgency of scaling up our ability to address climate change. So there's the ever so slight possibility that the stars may align for us, and that it may be that our timing is right, that we can learn from the mistakes of other campuses that made these investments maybe too early, and that we might actually be on track to do something that could be quite customizable to the things we care, that we care most about here. So with that, let me open for any questions you might have about either that research or the idea on a broader scale of Penn State's future research.
Thank you, Sally.
Yes.
Excellent. I'm monitoring the chat. If anybody has questions, wants to put in the chat box, go ahead and unmute yourself, or anybody in the audience.
I was wondering if the system has been looked at for a lot of the.
So the question is whether we've ever taken the system into a perennial. And we're doing that right now. And the perennial that we've chosen as our first target is a poplar for a couple of reasons. One of the reasons we put it into poplar is that we really wanted a tree. And that's only because I'm at Penn State now. I would never be doing trees were I not here. But if we can do this in poplar, then it opens the possibility we can do this to a lot of trees that aren't necessarily readily transformable yet, but we could put the time into getting them. So two answers there. We have MSH1 in poplar. We're evaluating whether it's working. With a perennial, the thing that's interesting to me is that they have a different utility of their meristems, I would say, because they don't necessarily flower each year. So it's not always a reproductive meristem you care about. Sometimes it may actually be a vascular cambium type meristem for a next season's growth. So would MSH1 alter that next season's growth as a memory the way that it would, in fact, through gametogenesis? So that's one of our key fundamental questions, is would perennials basically implement this differently? On an infrastructure basis, I can't really move forward, and neither can you if we don't have a plant transformation center on this campus. The fundamental problem being that there are transformation centers that are near us. Let's say Cornell has one, and Michigan State basically has something. Others have put it together, but they put it together for themselves. And what that means is that they haven't really put the energy and time into, for instance, tree transformation. And that's a whole research problem. That's not just a let's make sure we can do it problem. And I really think that there's merit in Penn State putting some serious time, money, and maybe even philanthropic investment into, let's just get it done. Let's find those transformation systems. And so this offers a really nice ready-made system for going right in. In other words, when people say, well, what would we transform with? Let's put in epigenetic resilience. Let's see what it does. And let's see what it does in a forest system. Because we have our own research forest here, we have the capability to do all of that. But I'm sure there will be other real great opportunities for transforming into trees once we have those systems in place. So poplars are our hope initially. Yeah.
I don't see anything in the chat either, so
Okay.
thank you, Sally.
My pleasure. Thank you for the opportunity. I'm very grateful.
Excellent job.